What are confocal microscopes used for? First of all, let's introduce you to a few types. There's the laser scanning confocal microscope, which uses a laser and illumination pinhole to get point-like light source illumination on the sample. The multiphoton laser scanning confocal microscope, which uses two lasers for a single excitation. And the spinning disc confocal microscope, which uses a disc with multiple holes in it between the light source and the specimen to shine a point-like light source. They all look pretty similar from these pictures and from the outside, but each functions slightly differently. No matter what type it is though, confocal microscopes are used to detect fluorescent labels on parts of a specimen of study. To get more information, we asked an NC State graduate student some questions about confocal microscopes.
So which ones of confocal microscopes are most frequently used, and is there a certain type that's easier for students to learn to use over another?
So most frequently and arguably better is the laser confocal. However, in my experience, the spinning disc is actually a bit easier to use.
In traditional wide-field microscopy, the specimen of interest is bathed in light from a mercury or xenon source. Then the image can be viewed by eye through the lens or captured on an image capture device. Using confocal, a more focused beam of light from a laser, for example, is scanned across a specimen. That small point of illumination is brought into focus with the objective lens and scanned by a computer. A photomultiplier tube, or PMT, detects the points of light through a pinhole. Because different parts of a specimen are labeled with different fluorescent probes, certain parts will light up when viewed with a confocal microscope, and the signal is interpreted in the PMT.
What are some of the applications that you use confocal microscopes for specifically in your work?
In some of my previous work, the biggest thing to do would be looking for specific proteins. So we would use antibodies that were attached to a fluorescent molecule, and those would bind to our protein in the cell, and then we could say, okay, this protein is in this portion of the cell at this specific time, or okay, our DNA looks like this in the cell right now.
How does it recognize the antibody? Because you said that it focuses specifically on that. How does it do that?
It's not that it focuses on the antibody per se,
Okay.
but the antibody has what's called a fluorophore conjugated to it. So on the end of an antibody, there's a little fluorescent molecule to work with.
Okay.
And so the antibody is going to bind your antigen, whatever that protein may be, and then the fluor, since it's attached to the antibody, will stay bound as well. And that way you have just the protein you're interested in lighting up.
It seems like adding fluorescent probes to all of your samples would be kind of complicated compared to just viewing the sample itself. So why do people use confocal microscopy? There are many benefits to using confocal microscopy. One is that there is a controllable depth of field in confocal microscopy. The goal is to eliminate any out-of-focus light in the viewing and imaging field so that only focus light and sharp light is imaged. This is why there is often a very shallow depth of field when using confocal microscopes, but this is good because clear images can be achieved. Additionally, many shallow depth of field images can be combined to formulate a 3D image of the specimen using a computer and different angles of laser light. So what kinds of specimens are imaged? Usually, the specimens of interest are either living cells or tissue that have been labeled with one or more fluorescent probes or dyes. The probes are conjugated to different parts of the tissue or different organelles or proteins in the cell so that the portions of interest can be viewed, or if the cell is living, certain parts of the cell can be tracked in real time.
What's the fourth dimension? You know this.
Time.
There you go. So the 3D is you have your 3D structure, so you're taking planes, you're collapsing them into a cube, and then you're doing that every 10 seconds over time. So all of a sudden you can say, okay, watch the entire cell change throughout time, if it's living.
Thus, the fluorescent dyes are useful in monitoring cell integrity, endo and exocytosis, membrane fluidity, protein trafficking, and chromosome analysis. Confocal microscopy has been used in ophthalmology, for example. In one paper, the researchers used confocal microscopy to calculate the nerve density in the cornea, shown in the upper two images. By interpreting the difference in fluorescence between the nerves and the surrounding tissue, they were able to do so. A computer program was then used to trace the nerves and calculate the density. In the bottom left image, researchers used confocal microscopy to visualize keratitis, or the inflammation of the cornea. The fungal filaments present in the eye in a person with keratitis show up as more reflective in the microscope. Thus, confocal microscopy can be used as a non-invasive way for diagnosis of diseases. In the bottom right image, fluorescent signals can be seen on different structural parts of the retina. Confocal microscopes have even been used to determine the thickness of the cornea based on the depth of the focal plane of the microscope. Confocal microscopes are widely used to characterize biological tissues. In another paper, researchers fluorescence marked certain parts of thyroid tissue on the left and colon tissue on the right and mapped the same type of tissue, but with cancer, which is at the bottom image in both types of tissue. Thus, one is able to see the shape and structure of the cancerous and healthy tissues using confocal microscopy. This is important because confocal microscopy can reduce the need for excision biopsies, reducing the need for pathological examination. Confocal microscopy has also been used to characterize colloids in certain areas of research, but this is not as common as is confocal microscopy in biological systems. However, it is still used for this purpose. The leftmost image is that of a reconstructed image of a colloid imaged using confocal microscopy. The right images are colloidal glass and gel, the top and bottom respectively. There are some things to consider when using a confocal microscope in order to obtain better images and 3D constructions.
Yep. So what is the most frustrating thing about using a confocal microscope?
It's not just the microscope itself. It can be kind of dim and hard to work with. The other problem is also going to be the imaging of whatever you're looking at. When it comes to laser confocal microscopy, you have a lot of small things you have to work with. So you've got the gain on each channel, your red channel, your blue channel, your green channel. Is the gain set correctly? If not, it can totally screw up your results. So you have to do each step very slowly. And slowly is another big thing. There are times when you can sit in front of that scope for 6 hours and not know what happens.
What does the imaging process entail?
It depends on your scope. There are multiple types of sensors and cameras and stuff like that. There is your basic camera, which is very similar to one of these guys, and some of your fluorescence microscopy will actually use a DSLR like that.
Okay.
But then there are very specialized ones, like the ones that's on top of our gel rig, the gel imaging thing we have in the lab. That one is a specialized CCD camera. Those are different because they've got bigger pixels and they're built to take up less light. So whenever you're doing certain kinds of things, you need that. Downside is, the biggest resolution you'll get out of those these days is still like 8 or 10 megapixels, which is small in comparison to today's cameras. So that's cameras. And then, usually with laser, you have something called a PMT, which stands for a photo multiplier tube. Because what's happening is, realistically, if you want to think about it, you've got a little laser, and it's hitting just this one point. And this one point is just emitting a tiny, tiny, tiny amount of light. So what happens is, this little photon comes up here, gets inside of the multiplier tube, and hits this, and that portion up here is going to bounce into 2, it's going to bounce into 4. So it's basically an amplification of the signal. The downside with that is, the more you amplify a signal, the more noise, background noise you get. So if you amplify too much, you're not going to be able to read anything. Your signal to noise ratio is going to be real bad.
Now we will take a look at how the axial focal plane and the scan line speed affect the content and signal to noise ratio of the image. Here we are looking at a pollen grain under a wide field microscope on the left and a confocal microscope on the right. As you can see, changing the axial focal plane varies the amount of focused light, more visibly evident in the wide field microscope on the left. In confocal microscopy, changing the axial focal plane does not visibly change the image's degree of clarity. However, it does seem to change the part of the pollen grain that is being focused on. Changing the scan line speed changes the grain of the image. A faster scan line speed results in a more grainy and consequently lower quality image, while a slower scan line speed results in a less grainy and higher quality image. Changing the red, green, and blue channels shows the certain color being increased more strongly, and more of that color is amplified and appears more vivid or brighter. Fluorescence emission gathered from the specimen is filtered through a confocal pinhole aperture to reject light that originates from regions removed from the focal plane. There are other factors to consider while taking an image with a confocal microscope. One of these is the spectral bleed, which is also called crossover or crosstalk. This occurs when one photomultiplier channel designated for a certain probe is recognizing other fluorescent probes as well. This complicates the interpretation of experimental results.
How do you remove the background from your samples?
Sometimes you can, but otherwise it goes back to that whole the gain thing. We talked about the more gain, the more grain,
Yeah.
right? But you can do another thing called dark field subtraction. And that's actually used in astrophotography as well. And basically what that does is you take an image without anything going on, and that image, you take 3 or 4 or 5 or 10 or 20 of those images, and that image kind of samples, okay, this is what the grain looks like when we're not looking at any kind of sample, right? So it takes that, and then it takes the image of the sample, and takes all that data from the previous 20 images or whatever it is, and subtracts, okay, if we take this average grain, subtract it from our actual image, here's what it should look like. Your biggest concern that you're going to run across is what you run across with every camera, is if you give that camera too much signal, at some point each photo site is going to fill up. And when that site fills up, your data is useless. Just like the overflow we saw in class the other day with some people. The fluorescence is so much that the camera can't read it because it's above its highest level.
There is also a consideration to make when choosing the probes you would like to use for your imaging. Certain probes work better with others, and it is important to choose the right ones in order to obtain the ultimate goal of an image with the best target signal and the minimal bleed-through artifacts, as discussed previously. Last but not least is the objective. This is an important consideration because it controls the information content of the image. The design of the objective determines the contrast and the resolution of the image, as well as the lateral extent of the image field in the microscope's view. There are many applications for confocal microscopy, including neurophysiology, neuroanatomy, stem cell research, photobleaching studies, DNA hybridization, bioluminescent proteins, and epitope tagging, in addition to the ones that were already mentioned in this presentation. Now you know a little bit more about the inner workings of confocal microscopy and what it's actually used for.